Good evening to everyone. I'm going to talk about parvoviruses and animal trial related to them. And since it is about a single pathogen, I approached it in the following way that I would like to refresh some information about parvoviruses and porcine parvovirus in particular. Then talk about some considerations for diagnosis, but we are in the fortunate situation that from Dr. Alberto we heard a very nice package of information. So I will go through on that part rather quickly. And then I will talk about the trial that we carried out with the parvovirus vaccine. So as we know, this is kind of a refreshment of the already existing knowledge. I just will highlight some interesting points. As we know, the parvovirus infect either vertebrates or arthropods, so they have a very wide host range. They are very small viruses, and very importantly, they do not have envelope, so quite resistant viruses, which is important for epidemiological point of view. And their genome is just around or below 5 kb. There's a paper entitled "That small doesn't mean simple," and this is very true for parvoviruses, as we will see it a little bit later. Considering the mammalian hosts, it includes humans, dogs, rodents, cows, and pigs. They have an icosahedral capsid, which consists exactly 60 copies of viral proteins. Among them, VP2 is the most important capsid protein because it determines the host range and it's a target for protective immunity. History demonstrates the range of parvoviruses. I'm trying to position the pointer, hope you can see it. And our main target, porcine parvoviruses, are a bit close to some rodent parvoviruses in close relationship and to the canine and feline parvoviruses based on the VP2 amino acid sequences. It's always important when you talk about phylogenetic tree to name the target. So now let's turn to porcine parvoviruses. They are called, how to say in the official way, ungulate protoparvovirus 1 right now. And I just put here this link for the International Committee on Taxonomy of Viruses, where one can follow the actual nomenclature. PPV is known since the late 60s of the last century, actually from Germany. And according to bioinformatic investigations, they have a common ancestor approximately existing in 250 years ago. And since then, there are 3 major groups that were separated at 150 and 110 years ago. And interestingly, they do not have geographic, how to say, proposition, so they can be found almost everywhere. We have to note that there are other genetic groups of PPVs, like up to now what we know about from 2 to 7, but their pathologic importance, at least to my latest knowledge, has not been established. So they might even be commensals or can take part in mixed infections. Parvoviruses as DNA viruses were for long considered very stable viruses from mutation and variability point of view, but it turned out that they are really variable viruses with a high mutation rate. And as usual, structural genes like VP2 is even more variable than non-structural genes like NS1. And it is just a small piece of information, but interesting that even among parvoviruses, there is a difference between recombination events taking place in their evolution or not. Up to now, PPV is considered to be free of recombination event. Now, if we just briefly go through the coded viral proteins, this small size does not equal with simplicity. Now it shows up here because it has two major open-ending frames, ORF1 and 2, but these open-ending frames code for a number of proteins that are being recognized lately or recently, because they are either coded for in an overlapping manner, or some of them are post-translationally modified products. And like this PLA2, this is a small protein that is very important for productive infection. And there is this SAT, small protein, that contributes to spreading capacity. And there is the NS1. NS1 has a number of functions. We will just a little bit talk about it later. Like NS1 is important for the productive replication, and it can be a discriminating factor between two major strains of PPV, like Cressy and NADR2. PPV has an expressed cytopathic effect, and it starts relatively early, 16-20 hours post inoculation or infection, and it can be seen clearly. I will show pictures about it just a bit later. And that is a very important factor, that it can establish latent infection in cell lines, and by this it can cause troubles, because if one uses contaminated cell line, if one uses it for virus neutralization, for example, the inherent PPV content might interfere with the result interpretation. So it can invalidate, or at least it can affect test results. For example, depreciate products. If it is present in products unwantedly, of course it is a problem. And of course it may change susceptibility to other infectious agents. They have different biotypes, as it is known. And if you see, just focus on NADR2 here and the Cressy strain, they are really remarkably different in pathogenic capacity, as you can see. And one important thing regarding parvoviruses and PPV, specifically, that very small changes, 1 or 2 amino acids, can have a dramatic effect on major biological functions like host range or pathogenicity. And it is known that PPV infection is influenced by the presence of other pathogens. Let's take a look at the epidemiology, at least on the most important factors. We learned it already from Dr. Alberto that it is widely spread and present all over. Very important, as I mentioned earlier, since it doesn't have an envelope, it is pretty much resistant, a hardy virus, and even resistant to quite extreme pH ranges and to heat. And since it is shed by a large amount of different excretions, it presents a continuous source for reinfections or new infections. But there are some chemicals that can be used with confidence to fight them. But it is an important factor that they can be almost everywhere, and they can be carried around by fomites, equipment, rodents, and as we learned, even by contaminated semen. Clinical signs will show up only in pregnant females, so we will see it later, and we heard already that it is related to reproductive parameters. And the important thing that even in the shadow of antibodies, the virus can circulate. So its presence cannot be excluded by vaccination. This graph shows very briefly some pathogenic aspects. What I would like to highlight is here that antibodies can be detected even as early as 6 days post-inoculation by AGI and VM testing. And an important thing, because you can see it later, of course, it stays in the slides, that abortion is not a prerequisite to recognize PPV infection, but the reproductive parameters or indices, but we already heard it before. This graph, I think, is interesting, especially in this late hour. It's almost a storytelling tale time. It shows that PPV, as the other parviruses, replicates in the nucleus, and it uses different mechanisms to get through the cellular membrane. And but after coming out from the nucleus, NS1 plays a critical role in causing apoptosis. One important mechanism is this reactive oxygen species. And another interesting thing, I just thought to show it to you, that the particle size, the virion size is so small, this approximately 20 nanometer, that if you take a look at the nucleus, it is all over with these nuclear pores, which serves as an entry for the viruses, for many viruses replicating in the nucleus. And parviruses just fall through these pores, while other viruses, DNA viruses like herpes virus, other virus, they have to uncode before this step. Parviruses, they fall through. And just coming back to this mechanism with ROS and apoptosis, is important because a certain parvovirus, a rat parvovirus, is used against glioblastoma in humans, and to my best knowledge, it is already over the experimental period, and these strains of viruses utilizing the similar mechanism, and NS1 plays a critical role in this aspect, and very tiny alterations like a single amino acid mutation makes this virus how to say functional against the cancer. It's just I think good to remember. You already know this the content at least of this slide, but I would like to highlight this milestone of the 70th day of pregnancy, because there is the borderline between immunocompetency and before that. So after that, the fetuses are able to produce antibodies against the pathogen. From diagnostic point of view, I couldn't really tell any newer information that you already know, just but maybe repetition is the mother of knowledge, or so to say, that the best is to submit fetuses when you suspect the presence of PPV. You show the pictures before. It is good to have, if there are, the range of fetuses from small to medium to large, and this is especially before 70 days of pregnancy. After that, one can go even for antibody detection from the body cavities, and even paired serum can be submitted to the lab. As we learned before, in the influenza related presentation, the diagnostic tools consist of direct and indirect tools. Here, in the case of PPV, we can have hemagglutination, immunofluorescence, PCR, immunohistochemistry, and even electron microscopy, immunoelectron microscopy. PCR is the most widespread, of course, and isolation. It is not a routine, but fortunately for PPV, it is not a very difficult thing. And now I would like to show you two pictures. This is a control cell, RPL2 cells. This is a swine kidney cell line. You can see just 3 days after the CPE, so it is just destroying the layer of the cells. And this is another picture that within 6 hours, between the 14th and 20th hour post-infection, you can see a dramatic effect, because the red staining means the the ready capsid, while the blue indicates the viral DNA, and you can see that it really kicks off within this period of time. Indirect diagnostic tools are the hemagglutination inhibition, ELISA, and VN. Actually, none of them are quite straightforward. I mean, ELISA, okay, is a standardized method that is easy to carry out, but the interpretation of the result is not too straightforward. For AGI, AGI is very sensitive to incubation conditions. It is a true factor, and VN as well, although there could be differences between vaccinated and the field challenge-induced titers. But VN is very sensitive, as I mentioned before, not only to the protocol, but to the reagents used, like contaminated cells, for example. There is a data that MDA's half-life is 20 days, and it lasts. The range is quite wide. It lasts between 9 to 22 weeks of age. And as it was mentioned before, it's good to highlight that antibodies can prevent clinical disease, but not the infection and shedding. For the prevention and control, there were historical ways, like when providing the aborted material to sows, but we choose nowadays option B, like in several aspects. And normally it is about revaccination of the breeding sows, but we heard about it before also. And now I would like to come to the trial and to show you some aspects why it was initiated. The 27A virus, as we will learn a bit later, so it got attention lately, and this tree comes from the latest issue of Disease of Swine book, and now we'll talk about the NADL2 virus, the Cressi, and the 27A-like strains. Actually, this tree is not the traditional tree because the scale here is set to time. This is excuse me, a very nice way, but not the usual way. What I highlighted here is that the tree was prepared out of the VP1 complex gene, and this is very important when you want to compare phylogenetic information. You have to stick to the same resources. Like if you go to this is a published tree, it's a different tree where you can see the grouping of the different PPV strains, and here on this tree, the 27A group falls into 27A virus, falls into group B. Yes, but this is about partial VP1, VP2 sequences. So this is not the same as we saw it before. And if you come here, if you take a look at these three, here, unfortunately, it was not specified which gene. It was viral protein gene. Most probably it should be VP2. Anyhow, but here, 27A falls to group V. But if you just go back, one can see that the grouping starts from up to down. So I consulted with the colleagues, so we consider it is kind of an arbitrary grouping. And if someone wants to really relate viruses, the best is to stick to the same region and specify if it is nucleotide-based tree or amino acid-based tree, and discuss afterwards. But the common thing in the 4 trees that all were scaled to time, and as we can see, it goes back to the 60s. The other one 60 years back. So I think it is quite in alignment with the statement previously mentioned at the beginning of the presentation, how the different lineages diverge. Now we come closer to our target, and when we wanted to deal with, or we got the task to deal with PPV 27A, we thought that it would be important to characterize it first, and then to develop an assay that could help us detect this virus specifically. Because of course one can recognize it by sequencing a good portion of the virus. Because we found out that the full-length VP2 protein gene, based the phylogeny on this part, we put the 27A strains nicely in the same group. Although you can see some minor differences here, but it was established that there are 5 nucleotide mutations that determine this group, the 27A group. One of them actually is silent mutation, so there is no amino acid substitution as a consequence, but the other 4 they are non-silent mutations. And importantly, they are dispersed, so it means that they are not next to each other. It is very difficult to target them by PCR, but we managed. So there is now a discriminating PCR, actually a duplex PCR that will tell the investigator if PPV1 is present in the sample or PPV1 and it is 27A at the same time. And now we just started using it for diagnostic purposes. So the objective was this trial to shed light on the belief or on the information that was spread all around that PPV27A is so pathogenic that and a new antigenic variant that traditional vaccines would not protect against it. And so we wanted to check Parvovirus against its main competitors, how it performs in a challenge trial. And we used a rather simple setup with the dumbbell periods at 6 months of age, screened before the trial and turned out to be negative for the virus by PCR and by ELISA. And we wanted to categorize the vaccines based on their content, on their virus content, PPV content. Now we show that it is the Cressy-like, but when we started the trial, we didn't know. So the first task was to characterize this virus. But we just go through the scheme. Here is the group of vaccines that we used, and all were used according to their SPCs. So there was nothing special. And we had these sampling milestones, the prime vaccination and mating, a challenge, and then at the end of the trial, a challenge was done at the 40th days of pregnancy, approximately, and after 50 days, the sows were sacrificed and the fetuses removed. They were evaluated for viability, like live, dead, mummified, for their length, for their weight, qPCR results, and isolation results based on published methods. The virus was a Hungarian isolate. It was just a few years before isolated in Hungary, fully sequenced, and it was found almost identical, at least on the VP2, identical to other established PPV27A strains. So now, if you go to the results, so first we wanted to establish the whereabouts of this of the virus strain of Parvovirus, and phylogenetically it grouped together with the Cressy strain. It was interesting because the general belief, and still in papers it is shown that all vaccines available nowadays commercially are NADL based, and NADL is here, you can see, and here are the two 27A strains that we are talking about. Then, if you take a look at this, at these capsomeres, each of them, ABC, represents a comparison between two strains, and the first is between K21, so this is the parvovirus virus strain, and the Cressy-like strain, and it is very easy to interpret, because the less changes you see, the more homology it means, because these differently colored dots represent differing epitopes. So they are important from immunological point of view. And if you see the others like 27A and K22 or NAD and 27A, there are much more differences than between Cressy and K22. So we took this relatively arbitrarily, arbitrary method that we named it Cressy-like strain. Now, if you go through the scheme, this is about serology. We used a commercial ELISA from an Ingezim ELISA, actually, and it was pretty much surprising that after vaccination, a very low portion of the samples crossed the positivity limit of the test. So you can see it here, and just a very few samples. And it was quite unanimous, except this group, but this group has the same viral strain in the vaccine as it is in the ELISA. So it is kind of an indicative of a stronger response to a homologous strain. But after the challenge, the titers reached a relatively high value for all of the groups, regardless of their vaccination status. VN virus neutralization showed the same, so moderate immune response, and there were no really major changes. The antigen we used, it was the challenge strain, actually. So to our surprise, serologically, it could not be made a big difference between them. So then we came to the beta parameters, and here the picture was much clearer and straightforward. Like parvovirus outscored all the groups. Like there was no dead or mummified fetus there, and you can read the figures because it was published actually, so it is available freely on the internet. I just would like to highlight some key effects that although there were a very low percentage of PCR positive fetuses, but isolation did not demonstrate live virus there, or the amount of the virus suitable for productive infection in cell lines. On the other hand, it was present in 13 and 36% of the fetuses, or a number, sorry, it is not a percentage, in one of the vaccinated groups and in the non-vaccinated controls. So our conclusion of this trial, which actually was quite a long trial, it is a very brief summary of this, that this so-called Cressy-like K22 based vaccine provided a stronger protection than the other NADR2 based vaccines, and it manifested in the number of sows with challenge virus affected fetuses and the different parameters of the fetuses. And based on the findings, 3 categories were set up or suggested, like full, sufficient, and insufficient protection based on the sero or PCR negativity of the fetuses. And the final thoughts that serological results or investigations did not correlate with vaccine protection. So therefore, our hypothesis is, and it is supported by literature data, that cell-mediated immune response and, in particular, strong immune memory should play a role in this strong protection. And until there is no adequate measure like serological or cellular test that is easily accessible and can be interpreted. So until its availability, the best way is to evaluate vaccine efficacy is the clinical protection. Of course, it would be important to find out the real prevalence of 27A virus, so a survey just would be useful for this purpose. We have already this method at hand, but we are pushing it forward. We would like to have a qPCR-based method. So this is being designed at the moment. And we would like to go further with the immunological characterization of these players like 27A, K21, and NADR2 by cross neutralization assays, because if they are carried out in the how to say good manner, they are really meaningful. So for that, yeah, we need some more experimental data. So with this, I'd like to thank for your attention, and I'm coming, luckily or not, or sadly or happily, with a single one question. And it reads, which one of the next statements is not right? Abortion is not a prerequisite in parvovirus cases. Antibody titers after parvo vaccination relate to protection. Besides the classical parvovirus strains, there are new ones. When infection takes place after the 70th day of gestation, piglets will be able to seroconvert. Specific antibodies can be detected. Thank you.